Hey everybody, in this set of videos I am going to cover the mechanism of crossing over that occurs between homologous chromosomes during meiosis. So before we can go over that mechanism, I need to review some concepts from one of our introductory biology courses and our genetics course. So essentially, let's start with a chromosome. Now, this is how I normally diagram a single chromosome. I don't think I normally make the centromere so big, but this is a single chromosome with a short arm here called the P arm and a long arm, which is the Q arm. Now, if this chromosome goes through S phase of the cell cycle, it will be replicated and it will look something like this. Well, conceptually, it will look something like this, and I almost colored the chromosome arm here. So this is also a single chromosome. And how do we know that? Well, because we only have one centromere here. But this centromere is holding together two sister chromatids. And the sister chromatids are identical, right? They should be identical because they were replicated during S phase from, the same template. So this is a single chromosome. This is a single chromosome. Single chromosomes can be made up of one chromatid or sister chromatids. Now, in diploid organisms, so diploid organisms are organisms that have two sets of chromosomes. One set is inherited from the paternal parent, the other one is inherited from the maternal parent. So if I should diagram, say, this chromosome over here, and this is a chromosome in a diploid organism, most cells, are going to have another copy of that chromosome. All cells except for the gametes, right? So we can say this one is inherited from dad, this one is inherited from mom. And these are homologous chromosomes. So two sets of chromosomes. This is one pair of chromosomes in the set, one from dad, one from mom. And this pair is considered to be a pair of homologous chromosomes. And they have the same genes at the same positions along the chromosome, but they might not have the same alleles, right? And what is an allele? So an allele is just a specific version of a gene. Now, okay, so if these should go through S phase, well, they'll look something like this. And here we have two chromosomes. They're the homologous chromosomes, and each chromosome has a pair of sister chromatids. And when these align, or when they're found together side by side during meiosis, they are called either a bivalent or a tetrad, which is essentially the same thing. Normally we call it a tetrad when we can see the four different chromatids that are part of this pair of homologous chromosomes. Another thing I want to touch on has to do with the cell cycle. And so you're probably tired of this cell cycle diagram by now, right? But here, let me diagram it briefly. So just a very crude diagram of the cell cycle. And M will be for mitosis. We have G1 phase, S phase, G2 phase. All of these phases are part of interphase. And even though I tend to diagram the chromosomes in their condensed forms here. So this is a condensed chromosome. Chromosomes are only found in the condensed state, right, during mitosis or meiosis. most of the time they're decondensed, they're unwound. And a major area of research is involved with trying to understand how chromosomes are arranged in the nucleus during interphase. And it's thought that each chromosome has its own territory. So chromosome territories. And so they would be unwound. And let's say chromosome 1 would be over here. And maybe both chromosome 1s would be over there in that section. Chromosome 2 would be over there in that section. And so on and so forth. So this is a very important area and current area of research. So it's not like they're just randomly arranged in the nucleus and they're unwound and tangled, but there is thought to be some organization to how the chromosomes are arranged in the nucleus during interphase. And I really just wanted to point out that, you know, I always diagram chromosomes like this. I don't want you guys to think that, okay, they're always condensed. No, normally they're decondensed and arranged in their territories in the nucleus during interphase. And then they will condense during mitosis. But we diagram them like this because it's, you know, easier to explain or talk about chromosomes when we have a nice little neat diagram like that. Now, another important point is we need to just touch on a major difference between mitosis and meiosis. So specifically metaphase of mitosis and meiosis. And meiosis has two metaphases, right? So metaphase 1 is what I'm going to touch on. So during metaphase, in mitosis, the homologous chromosomes, all of the chromosomes, line up one by one on top of the other on the metaphase plate. And we have the microtubules attaching to the centromeres. So these are, let's say this is the pair of homologous chromosomes I diagrammed on the previous sheet of paper. Now they'll line up one on top of the other. So during anaphase, the next stage, one sister chromatid from each of these chromosomes, you know, goes in one direction and the other one goes in the other direction. And the reason that occurs is that the purpose of mitosis is to make two genetically identical nuclei, you know, from a parent nucleus. So from one nucleus is, you know, the purpose of mitosis to make two genetically identical nuclei. Now, during metaphase 1 in meiosis, those same homologous chromosomes, that same pair of homologous chromosomes, would line up side by side. So somehow those chromosomes have to find each other, and then they'll line up side by side on the metaphase plate. And let me color this one in over here. And so these are homologous chromosomes. One is attached to one microtubule, the other one is attached to another microtubule, and they will go in opposite directions during anaphase 1. And the reason this happens is the purpose of meiosis, so one of the purposes, is to reduce the genetic content by 1/2. So with mitosis, the purpose is to make two genetically identical nuclei. Meiosis, the purpose is to reduce the genetic content by 1/2. So in the gametes will only have half of the genetic content of the normal cells in the organism. Another purpose of meiosis, or of this, is, well, one thing is, so this thing right here, this overlap, what is that? So that is the cytological manifestation of a crossover. Cytological, what is that? That's a fancy term, means it's what you see when you look under a microscope. So when we look under a microscope, we see an overlap between arms, two of the sister chromatids of the homologous chromosomes. We see an overlap. We're going to call that's called a chiasma. And what the chiasma is, it's what happens is what we see when a crossover has occurred. That's what we see when we look under a microscope and see a tetrad here, a pair of homologous chromosomes. We see an overlap of the sister chromatids. This is a chiasma, and what it means is a crossover has happened at that location. Now, we need at least one chiasma to form between each pair of homologous chromosomes because it holds these together during metaphase 1. It holds the homologous chromosomes together. It also allows for crossing over.